another very important subtopic. We discussed that there are two types of enzymes. Okay? One of them is intracellular and the other one is extracellular enzyme because the question is of this. So these were certain traits that we did yesterday for enzymes. Usme se one of them is intracellular and extracellular. So this is a very repeated question again. Okay, they give you this question, okay, any enzyme which is working inside the cell, this is known as, so any enzyme which works within your cell is known as intracellular. And any enzyme which works outside of your cell, it is known as extracellular. Okay? So this is the first question again in your past papers. Okay, hydrolytic enzymes can function within the cell. Okay? They can function within the cell or can be secreted by the cell where they're able to catalyze reactions. State the term used to describe an enzyme that functions within the cell. So what am I going to write?
Intracellular.
Intracellular.
Intracellular. ठीक है? So कभी they give you intracellular and कभी they'll give you extracellular. So this was the one thing that my teacher did not tell me, and I used to make mistakes again and again. Okay, if they specify this thing, okay, intracellular enzyme and extracellular enzyme, and then your life will become easy for 1 mark question. So this is a question for paper 2s and for paper 1s as well. Now moving to the other important concept, which is Michaelis-Menten constant. So before explaining this question, I'll explain you what Michaelis-Menten and what Vmax is. Okay? So Vmax means velocity at maximum, which means that your enzyme is working at the maximum velocity. And Vmax is like the last terminating point of your enzyme. So basically this line, which is now, you can see the termination over here, right? So the termination line is known as Vmax. So this is my Vmax, that my enzyme is working at the maximum velocity over here. Okay, so this is my Vmax. And how do I find its Michaelis-Menten constant? I find it by, so if, for example, 1.0 is my Vmax, then I'll divide 1.0 with 2. So yeah, I'll divide it into half. 1.0 ka half is going to be, let's test your maths. 1.0 ka half is
0.5.
0.5. So somewhere around here, we'll get 0.5. Okay? So pehle what I did, I found out the Vmax value. Usko I did half of it. Now I'm going to find out the Km value, which is the constant value. So since 0.5 is at y-axis, I look at x-axis and then I'll go down here. So if my value is 5, then 5 is going to be my Km value. Then again, agar dusre wala reaction ke liye, if I see my Km value, then somewhere around this line, I'm not sure ke ye hai, exact nahi pata, but kuch isi tarike ki value is going to be our Km value for the other reaction, for the other enzyme. ठीक है? So this is your enzyme G ka Km value, and this is enzyme H ka Km value. And what exactly Km value represents? Usman, do you want to take the lead over here?
Miss, they represent the affinity of enzyme and substrate concentration.
Exactly. Yeah. So higher the Km value, that means, okay, higher Km value means
Less affinity of enzyme
the
and
lesser
substrate.
the affinity, which means okay, less enzyme substrate binding will occur at this point. Okay, and a vice versa situation. You can also find it in my slides. Less affinity. So let me show it again over here as well. Yeah, it's written over here. It's basically written how you find out the Michaelis-Menten constant from Km. So it says that higher the Km value, lower will be the affinity of enzyme and substrate, and vice versa is the situation. Okay? So this is exactly how you are going to attempt this question. And this is also one of those questions, one of those most favorite questions of Cambridge. Okay? So be mindful when you're doing this. The two enzymes have different values of Michaelis-Menten constant, Km. Use figure 3.1 to derive the Km value for enzyme H. Show your working. So you need to write the mathematical values over here. Say yeah, you do not need to write we took the half of Vmax and the stuff like that. Do not write English, just show your working. So you need to find out the Km value for enzyme H. What are you going to do? We already found out the half. 0.5 is your Vmax. So you'll write Okay, Vmax ka half is at Vmax is like 1, and when you divided it with half, you made it half, it became 0.5. So you'll write 0.5 AU. AU is arbitrary units. Okay? Make sure that you write units. It's important. AU, 0.5 AU. Now you'll go at 0.5 and then you'll search for the substrate concentration. So substrate concentration over here, I believe, is somewhat 8. Yeah, it's 8 8 m mole dm cube. ठीक है? So your
Yes.
substrate, your basically your Km is Km for enzyme H is 8 m mole per dm cube. Acha. With reference to figure 3.1, explain how the values of Km for these enzymes provide information about the relationship between the enzymes and their substrate. Again, the same thing. So I just mentioned enzyme. Ab aap yahan pe you can give a solid comparison in terms of values. You can find out the Km of enzyme G and you already know the Km value of enzyme H. Enzyme H is 8 and again, tentatively we look for enzyme G. It's somewhere around 4. Yeah, 4.1, 4 point something, something around 4, right? So you can write K enzyme G has low Km. For acha, starting off, you need to mention कि Km exactly hota kya hai. So you'll write Km is the affinity of enzyme for its substrate, and then you'll write कि G has low Km value. So which means that enzyme G has a higher affinity for its substrate than H. ठीक है? I'm also writing it over here. Km shows the affinity. First point is that I'm explaining what exactly Km is. So Km is affinity of enzyme with its substrate. First point, which means this is my first mark of enzyme with its substrate. And then you'll write enzyme G has low Km value, has low Km, which means it has higher affinity with its substrate. Okay, so I'll get 2 marks on writing these pointers. Higher affinity. उस्मान, मिन्हा, संगीन सबको समझ आ रहा है?
Yes, miss.
Yes, miss.
Okay. अगर lost हो गए तो फिर भी बता दे. We'll do it again. अच्छा जी, so this is very easy. Name the vesicles in the cell that contains hydrolytic enzymes.
Lysosomes.
Lysosomes. इतना confidence से जवाब आया ये वाला. Lysosomes. I appreciate when you guys respond nicely. Okay. State one feature involving mitochondrial structure or function that could lead to the release of these internal signals. बात क्या हो रही थी? The cells has internal chemical messengers that signal when an old or damaged mitochondrion should be broken down. Suggest one feature involving mitochondrial structure or function that could lead to the release of these internal signals. How would we know mitochondria is not it's not like performing well or it should now be broken down? How will we know? How, not exactly we, how will the cell know?
ATP कम होगी.
ATP की प्रोडक्शन विल डिक्रीज, नो हो जाएगी या बिल्कुल ही लेस हो जाएगी, या माइटोकॉन्ड्रिया की रेप्लिकेशन विल स्टॉप अकरिंग बिकॉज वी नो दैट माइटोकॉन्ड्रिया इज अ सेल्फ रेप्लिकेटिंग ऑर्गेनल. So we'll see कि यहाँ पे no ATP will be produced and we'll see कि ATP is not being replicated. ठीक है, कोई भी pointer, anything which you like. No replication. And why I'm talking about replication? Because I know mitochondria is a self-replicating structure. ठीक है, आर वी गुड? समझ आ रही है?
Yes.
जी।
अच्छा जी, so now this is something else. Let's go to P1. Okay, so अच्छा, um अच्छा, did I mention that we'll take three chapters test all together? This is like the second chapter. We'll take the test of biological molecules, enzymes, and mitosis in like the upcoming weeks. Next week we'll do mitosis and then uske past papers और उसके बाद so start preparing biological molecules, enzymes, and next week में फिर mitosis शुरू हो जाएगा. All right. So just a reminder के be prepared for your test. I think 2 weeks में होगा वो. So, first question is, a student investigated the effect of enzyme concentration on the rate of hydrolysis of proteins in milk. When the enzyme and milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. The student investigated 5 different enzyme concentrations and reported the time taken for the mixture to become clear. What is an appropriate control for this investigation?
A.
Carrying out an experiment where the enzyme solution is replaced with water. Why A? Why should हाँ, go ahead.
concentration now.
कोई concentration ना हो, ठीक है. So this is A. Perfect. What determines the specificity of an enzyme? ये तो बहुत आसान सी चीज है. A covalent in the other bonding between R groups of the polypeptide. And why are they talking about polypeptide? Because enzyme is protein in nature. Determine the active site की shape. The covalent peptide bond between the amino acid of the polypeptide. The covalent peptide bonds between amino acids of the polypeptide. Mein, do we have covalent bonds or do we just have peptide bonds?
Peptide bonds.
Peptide bonds. So this is also a useless option. Hey, the shape of the substrate molecule. The shape of the substrate molecule, will this determine कि enzyme का क्या specific shape होना चाहिए? No, right? वो तो एक दूसरी चीज है. That is a part of some sort of reaction. ये तो determine नहीं करेगा ना कि enzyme जब बन रहा है तो उसकी क्या specific shape होनी चाहिए. So the answer should be C, one only. That covalent bonds, R group के covalent bonds and other bonding polypeptide की. तो यहाँ पे they are now talking sense के between R groups of the polypeptide, यानी इसमें covalent bonding भी होगी. ये primary structure की बात हो रही है, जो कि primary structure जो कि first stage होता था. Now since we are done with biological molecules, we now know कि primary structure for secondary structure जो है वो आपका बना रहा होता है प्रोटीन को वो डिटरमाइन कर रहा होता है, राइट? सो this is like the pointer responsible to determine the shape. ठीक है?
जी।
अच्छा, मार्क दिस क्वेश्चन इज वेरी इंपॉर्टेंट. ये वो क्वेश्चन है जो आपको हर साल आपके पेपर में मिलेगा कि Vmax और Km की वैल्यूज पे क्या अफेक्ट आता है इन प्रेजेंस ऑफ अ कंपेटिटिव इनहिबिटर. दिस इज एब्सोल्युटली वेरी 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 इंपॉर्टेंट. 4-5 स्टार्स बना के अपने पास स्क्रीनशॉट करके रख लें इसको, बिकॉज ये बहुत दफा आता है. एंड अच्छा, क्या कह रहे हैं कि Vmax and Km are determined for enzyme catalyzed reaction. What will be the effects on the Vmax and Km in the presence of competitive inhibitor? Competitive inhibitor क्या करता था? It's a reversible reaction and it has the same shape as that of substrate. So वो भी जो है वो आपके active site के साथ bind कर सकता है. So if for example your competitive inhibitor binds with your enzyme, so will there be any impact on your Vmax? Because this is a reversible reaction, there will be no impact on the Vmax. कभी आपका inhibitor आके bind करेगा, कभी substrate आके bind करेगा. Substrate की concentration बढ़ा देंगे, तब भी reaction होता रहेगा. और competitive inhibitor एक दफा bind करता है, reaction नहीं होता, वो remove भी हो जाता है. ठीक है? But your Km will increase. Why will your Km increase? Because यहाँ पर affinity जो है substrate के साथ in presence of competitive inhibitor कम हो जाती है. हमने यही पढ़ा था ना कि अगर एंजाइम है उसकी शेप जो है वो सेम होती है सबस्ट्रेट की एंड योर कंपैटिटिव इनहिबिटर की, तो ये दोनों सेम शेप के होते हैं। अब इफ इट बाइंड्स विद योर एंजाइम देन अगर सब एक अगर इनहिबिटर आके बाइंडेड विद योर एंजाइम then Km will increase definitely because Km represents the affinity between enzyme and substrate, not enzyme and inhibitor. So it should definitely increase while your Vmax will remain the same because आपका जो रिवर्सिबल रिएक्शन है तो आपका कभी ना कभी तो आपका बाइंड हो जाएगा ना सबस्ट्रेट, रिएक्शन तो प्रोसीड करेगा। ऐसा तो नहीं है दैट योर रिएक्शन डिड नॉट टेक प्लेस, योर रिएक्शन विल कीप ऑन टेकिंग प्लेस एट द सेम रेट। ठीक है, so this is 3D. Your Km increases, your Vmax remains the same. Okay. So this is again अच्छा question है, conceptual question है rather. So it's saying that the graph shows the effect of temperature. It shows the effect of temperature on the rate at which enzyme in a biological washing powder digests and removes fruit juice stains. Which statement explain the shape of the graph at temperature higher than T? तो ये तो बहुत आसान सी बात है, तो आप लोग कर लोगे। Bonds are broken between the R groups of the amino acids
B.
in the B,
C.
C. All right, just let me see. The tertiary is altered, yes. One is correct. There are more collisions between the enzyme and substrate. Two is wrong. The shapes of the active site and enzyme are no longer complementary. So yes, 1, 3, and 4. This is C. ठीक है जी. All right. The graph shows how the concentration of components 1, 2, 3, and 4 of an enzyme catalyzed reaction changes with time. Which row identifies the components of this reaction? 1, 2, and 3, 4. You need to identify three of all four of them. ये best किस चीज को जो है वो represent कर रहे हैं. So I believe that your component 1, as your time is increasing, the concentration of your product should increase, right? So 1, I believe, should be your product because as your time increases, your product increases as well. Then your component 2 should be your अच्छा एक मिनट, let me identify 3. It's easy to identify. 3 क्या लग रहा है? 3 is very simple to identify. 3 ये रहा. एक चीज बढ़ रही है, इसका इनवर्स है. 1 का इनवर्स is 3. That one of the things is in
अनबाउंड एंजाइम कम हो रहा है सी शायद.
3.
में सबस्ट्रेट कम हो रहा है।
सबस्ट्रेट कम हो रहा है, एग्जैक्टली exactly. आपका एंजाइम कम नहीं होता ना, एंजाइम तो आपका सेम रहता है, इट रिमेन्स अनचेंज्ड, राइट? So 3 is your substrate, that as the time is progressing, substrate is decreasing and product is increasing. आया समझ?
जी.
So now we need to look for, let's do it with some other color. Confusing हो रहा है यहाँ से. एक minute. This is your product and this is your substrate. 3 is your substrate. Acha ji, now what we need to identify? We need to now identify 2. Okay, so this is ये तो same color आ रहा है. Okay, so this is 2. We need to look for 2. 2 क्या लग रहा है? 2 की concentration initially ज्यादा है, फिर कम हुई, फिर थोड़ी ज्यादा होना शुरू हुई है. So वैसे तो you get your answer after identifying your 2 options, which should be D. Because component 1 you identified it as product, you identified component 3 as substrate. So हो रहा है, कभी फिर बाइंड हो रहा है, ठीक है? तो उसकी जो बाउंडिंग एबिलिटी है वो 2 से रिप्रेजेंट हो रही है. एंड फिर 4 जो है इट्स योर एंजाइम सबस्ट्रेट कॉम्प्लेक्स, बिकॉज वो एक चीज बढ़ रही है एक चीज कम हो रही है, तो एक चीज का इस तरीके से शो होना भी इम्पोर्टेंट है टू शो के यहाँ पर जो है वो अभी भी रिएक्शन चल रहा है, बिकॉज आपकी प्रोडक्ट भी अभी जीरो, आपका सबस्ट्रेट अभी जीरो नहीं हुआ है. तो इसका मतलब है कि यहाँ पर अभी आपका कॉम्प्लेक्सेस आर स्टिल दे आर बीइंग मेड. 6. Which statement about the active site of an enzyme is correct?
A, miss.
A.
A. About the active site of an enzyme is correct. It always has a specific fixed shape. Usman, you were present in yesterday's class. How can you say that it always has a specific fixed shape? We talked about the induced split mechanism, right? Where we mentioned that active sites are always, they're not always specific. Sometimes they do induce a conformational change. कि जब उसमें एक ऐसा substrate आता है तो it's similar होती है पर आपका conformational change induce हो रहा होता है induced split mechanism में तो हमेशा they do not have specific fixed shape, right? उन्होंने कहा है it should be correct. So it reduces the total energy of the product. This is wrong. It does not form chemical bonds. ये भी wrong है. It is determined by the primary structure of an enzyme. The shape before the proteins are made, their things are determined by the primary structure, by primary level organization, right? So this is D. Energy. So we'll see you next week on Saturday, inshallah, and we'll do few past papers of enzymes again, and then we'll start with mitosis. Okay? So mitosis is a slightly technical chapter, and try to attend the live classes of mitosis because from now onwards, from mitosis, your syllabus will get tougher and tougher. It's more technical and it's not going to be easy anymore. We have mitosis, we have nucleic acids, we have transport in plants. फिर आपका cardiovascular system, it's completely different from what you studied in O levels. Not completely, but बहुत technical aspects हैं. So try to be live in the classes. I'm talking specifically to those जो recording वाले बच्चे हैं. So yeah, this is it from my side. ठीक है, तो take care and jazakallah khair for joining.
Allah Hafiz, miss.
Allah Hafiz, miss.